Atoms, ions, molecules, and the interactions between them are essential for life. Living cells are all composed of the same chemical elements. The functions of atoms, ions, and molecules in cells depend on their shapes and chemical properties, particularly the number and arrangement of protons and electrons. Electrons have different energy levels, which cause them to follow different orbital paths around the atomic nucleus and leave certain electrons available for bonds to form. So what does this look like? Electron arrangement, we're going to be focusing on a few key elements, and we use the acronym CHNOPS to describe these elements. These 6 elements are going to be the most common substances that we find contributing to molecules in living organisms. So here's what the electron arrangements for those elements look like. So we have carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And if you look at these on a periodic table, you'll notice that these are the non-metallic and non-noble gas elements that are in the simplest level, so the first few levels of the periodic table. What this means for their atomic structures is that there's not a whole lot to them, particularly things like hydrogen. We're looking at 1 proton and 1 electron contributing to this atom. And also critical importance here, this means there aren't a whole lot of different electron orbitals that we need to deal with in looking at how these atoms interact within molecules. So you can see here, hydrogen just has 1 electron and 1 orbital. Carbon, nitrogen, and oxygen all have 2 levels of electron orbitals. They're not full. And phosphorus and sulfur have 3. The not full descriptor describes to the outer orbital for all of these elements. If you look at the outer orbitals, the first orbital can hold 2, the second can hold up to 8, and same for the third. And you can see none of these has a completely full valence electron orbital. This is going to be really critical because all of these atoms are going to try to stabilize and reach a stable valence electron level by sharing or exchanging electrons. Okay, it can be really tempting to stick with this 2-dimensional structure of atoms and think about them only that way when we look at them in molecules, but do keep in mind, and we'll talk about this more later, that they actually have a 3-dimensional structure. So this carbon, its 4 outer electrons here correspond to the 4 bonds that it's able to make in this molecule that's represented here. Differing electronegativity of atoms results in different distribution of electrons in a bond. Covalent bonds are based on electron sharing, not charge. Covalent bonds can be single or double, depending on how many electrons are shared, and polar or nonpolar, depending on how evenly electrons are shared. Ionic bonds, hydrogen bonds, and van der Waals attractions form from positive to negative interactions. Chemical reactions rearrange electrons to form new bonds between reactants. So this idea of electronegativity is going to be very important as it contributes to polarity, and this is something we'll be coming back to. So if we look at electronegativity, this is a way of describing how the nucleus of an atom pulls on the electrons around it. So a nucleus that has more protons in it relative to the number of electrons in its valence shell is going to be more electronegative. So of our CHNOPS elements, here you can see represented, hydrogen has the lowest electronegativity, whereas phosphorus and sulfur, because they have a much larger nucleus, have a higher electronegativity. So this means when each of these atoms participates in a bond with another atom, it's going to have a proportionate pull on electrons in that bond. And this controls what type of bond is actually able to be formed between those atoms. So in some cases, the electrons are shared between the atoms. So you can see here we've got 2 chlorine atoms, and they are both sharing equally because they have the same electronegativity. They're the same element. In some cases, the sharing is not quite even, but it's not so uneven that the electrons completely dissociate. And this is what we call a polar covalent bond. So when chlorine bonds with hydrogen, the chlorine has a much higher electronegativity, so it pulls much more strongly on the electron that hydrogen is contributing to that bond. That means the electron spends more time near the chlorine and less time near the hydrogen. So the hydrogen doesn't have a full charge, but it picks up a partial charge. That's what the little delta symbol means. And conversely, the chlorine picks up a partial negative charge. When we have really big differences in electronegativity, this is when we get transfer of an electron. And this is an ionic bond. Ionic bonds are actually really rare in the context of cell biology. What we instead see is ions, because when you place an ionic bond into water, which is a significant amount of what's going into a cell, the water helps to dissociate these two charged atoms from each other. Since the sodium here no longer has its electron, it's become positive. The chlorine has become a negative chloride ion. So these can be easily pulled apart when they are placed in water.